Hello, and welcome to Contact's mini webinars, where we're going to discuss today what are reasonable targets to test for. My name is Dr. Faith Critzer. I'm an associate professor at the University of Georgia in the Department of Food Science and Technology. I'm a member of the Contact team, and I'd like to remind you that this work has been supported by the Specialty Crops Research Initiative from the USDA National Institute of Food and Agriculture. Any opinions, findings, conclusions, or recommendations expressed in this presentation are those of myself and do not necessarily reflect the view of the U.S. Department of Agriculture. One of the first questions we regularly get is, are there any regulatory requirements for testing product? To be brief, which I know is rare, the answer is no. Unlike with meat and poultry, which are regulated by the USDA, where there are product testing requirements in their regulations, there are no product testing requirements in the produce safety rule. Any entity identified as a farm will fall under this regulation. For entities which handle raw produce for further processing or packing that do not fall under the produce safety rule, the preventive controls rule may apply. Under this rule, operations may elect to conduct product testing for indicator organisms or pathogens as a part of a verification activity to show that their food safety plan is working as intended. Even though there is no regulatory requirement to test product, there can still be some very good reasons why operations may want to consider this strategy, as Michelle Danieluk has already mentioned in one of our other Contact mini webinars. Or you may have buyer or marketing agreement requirements which call for product testing. So how do you choose what to test for? What to test for is as important a question, and the answer is going to depend on what question you're trying to answer or the overall goal of your testing program. Let's start with indicator organisms. We think about these in a lot of different contexts beyond testing product, but for our purposes today, let's focus on this use. With most indicators, we are looking to enumerate or count the number of microorganisms present in that group. First is the aerobic plate count, which also goes by the standard plate count or total plate count. The aerobic plate count doesn't indicate food safety risk, but can help establish shelf life and determine product quality. But again, since it's not related to safety, let's go ahead and take it off the table. That leaves us with three target indicator organisms that are interconnected. Enterobacteriaceae are a taxonomic family of bacteria that represents more than 50 genera of bacteria, with some examples listed in the blue square. These organisms are widely found in the environment and can be found in water as well as terrestrial environments. Coliforms are a subset of organisms within the Enterobacteriaceae family, which are defined by growth characteristics to separate them from other members of this family. These characteristics include that they ferment lactose with the production of acid as well as gas when they are incubated from 95 to 98 degrees Fahrenheit. The organisms represented in the coliform group are also widely found in the environment and commonly found on fresh produce. Escherichia coli, which is usually shortened to E. coli, is a specific organism that is part of the coliform group and even larger Enterobacteriaceae family that is highly associated with fecal contamination. E. coli also survives in the environment fairly well. Given that fecal contamination is a primary source for foodborne pathogens, we have a better linkage with populations of E. coli and probability of finding a foodborne pathogen. If we were to test the same product for all three groups, we would expect the largest populations on total Enterobacteriaceae assays and coliforms, followed by E. coli. Why would you select an indicator organism when testing product? The first reason is to verify food safety practices implemented at harvest, packing, and holding are effective. If you tested in the field and after packing and the populations increase from what they were originally, this indicates a contamination source at some point during handling and an underlying problem that needs to be fixed. E. coli may also be used to indicate increased probability of foodborne pathogen contamination, but this relationship really needs to be established and will differ by crop. In this approach, quantifiable limits could be established, as well as presence or absence of E. coli. with quantifiable limits being more commonly applied since populations of E. coli are expected in raw produce given that they survive in the produce growing environment. Moving into foodborne pathogens, when considering what to test for, Salmonella, Shiga toxigenic E. coli, and Listeria monocytogenes are all examples of bacterial foodborne pathogens which have historically been associated with fresh produce outbreaks. The USDA mandates testing for certain meat products for 7 serotypes of Shiga toxigenic E. coli, which are the largest causes of illness within the US. Serotyping is another way microbiologists differentiate microorganisms that are very closely related. In this example, we are typing the somatic antigen, abbreviated as O, which is present on the surface of Shiga toxigenic E. coli. Regulations were first established for E. coli 0157, but have since been extended to the next 6 serotypes causing the most illness, many times referred to as the Big Six non-0157. This has spilled over into testing methods and a myriad of rapid molecular tests which can be performed to differentiate by virulence genes as well as serogroup for these 7 serotypes of Shiga toxigenic E. coli. Regardless of what you call them, all of these variations are deemed to be a pathogen and thus an adulterant if found in a product. If products test positive for a pathogen, it is considered adulterated and cannot enter commerce. If it is already in commerce, a recall must be initiated. As to which pathogen or pathogens you may want to test for, historically, we will look for which pathogen commodity pairs we have seen in past outbreaks. When thinking about why you would select a foodborne pathogen for testing product, there may be some very good reasons that we've discussed in other talks in this series for testing product, such as a verification activity, to make sure farms' food safety plans and good agricultural practices are being effectively implemented. It should be reiterated once more that the most rigorous sampling plans will likely not detect the pathogen contamination that is sporadic and at low frequency. So don't let it give you a false sense of security. You may also elect to test product if you're trying to determine when and how product may be contaminated as part of a root cause analysis, or to answer other specific questions so you can take steps to remedy the situation. So in bringing it on home, I would like to thank you so much for taking time to learn more about testing product for indicator microorganisms and pathogens. The most important things to keep in mind is what questions you're attempting to answer. That should really drive what you will test for ultimately. Also, understand the limitations that we've discussed for different testing options, what results mean and what they do not mean. Thank you.